Far silent depths. Mariana Trench marks the deepest known part of Earth's ocean, stretching through western Pacific near Guam. Far silent waters. Its lowest area, Challenger Deep, drops farther below sea level than Everest rises above it. Far silent pressure. This trench forms where Pacific Plate dives beneath smaller Mariana Plate at a subduction zone. Far silent darkness. As one plate sinks and seafloor bends downward, creating a long curved scar across the ocean. Far silent trenches. From space, the trench looks invisible, but below waves it slices dramatically into Earth's crust. Far silent currents. Most of Trench lies east of the Mariana Islands, an arc built by tectonic collisions. Far silent seafloor. Scientists measure the deepest points carefully because pressure currents and terrain complicate every estimate underwater. Far silent abyss. Even today, exact depth values vary slightly depending on sonar methods, instruments, and calibration standards. Far silent silence. At the bottom, pressure exceeds 1,000 times atmospheric pressure felt at Earth's surface normally. Far silent worlds. That means every square inch experiences crushing force strong enough to destroy ordinary unprotected machines. Far silent shadows. Sunlight disappears long before the trench floor, leaving a black world without natural daytime illumination. Far silent frontiers. Temperatures stay cold, usually just above freezing, although geothermal activity can locally alter conditions somewhat. Far Hadal depths. Food is scarce this deep, so life survives on marine snow carcasses and microbial chemistry. Far Hadal waters. Marine snow includes dead plankton, fecal pellets, mucus, and drifting fragments sinking from brighter waters. Far Hadal pressure. Occasionally, a whale fall delivers a huge food bonanza to deep scavengers and bacteria below. Far Hadal darkness. The trench is not empty. It hosts amphipods, microbes, sea cucumbers, and Hadal snailfish today.
Far Hadal trenches. Hadal zones describe ocean depths deeper than 6,000 meters, including trenches and deepest troughs.
Far Hadal seafloor. Life here adapts through flexible proteins and specialized membranes and chemistry that resists extreme pressure damage. Far Hadal abyss. Some deep animals avoid gas-filled spaces entirely because compressible cavities fail under enormous surrounding pressure. Far Hadal worlds. Researchers found snailfish near the trench bottom, proving vertebrates can live astonishingly deep underwater today. Far Hadal shadows. These deep-sea snailfish appear delicate, yet their bodies contain molecules helping proteins keep functioning under pressure. Far Hadal frontiers. Large amphipods roam the Hadal floor, scavenging whatever organic matter reaches the abyssal sediments below. Far shadowed depths. Xenophoever's giant single-celled organisms can carpet muddy slopes and provide habitat for smaller animals today. Far shadowed waters. Microbes also thrive there, metabolizing chemicals and recycling nutrients within sediments hidden beneath darkness today. Far shadowed pressure. Some bacteria consume organic matter, and others exploit compounds released by rocks' fluids or decomposition below. Far shadowed trenches. Each layer of mud can record tiny fossils, ash, or contaminants delivered across decades today. Far shadowed currents. Scientists sample these layers like pages inside a buried archive of ocean and planetary history. Far shadowed seafloor. Trench also matters for earthquakes because subduction zones store stress and sometimes release devastating ruptures. Far shadowed abyss. Nearby tectonic activity can trigger quakes and tsunamis, though not every trench segment behaves identically. Far shadowed silence. Volcanoes west of the trench arise when water from the descending plate helps generate magma. Far shadowed worlds. So the trench connects directly to the fiery island arc above, despite its frozen darkness. Far shadowed shadows. This pairing shows how Earth's deepest ocean and volcanically active islands share one tectonic engine. Far shadowed frontiers. Humans first reached Challenger Deep in 1960, aboard the bathyscaphe Trieste, a remarkable descent. Far abyssal waters. They reported a flat, silty floor, surprising many who imagined jagged cliffs and constant chaos.
Far abyssal pressure. Decades later, robotic vehicles returned with cameras, sensors, and samplers more capable than Trieste today.
Far abyssal currents. Their missions revealed ridges, sediment plains, escarpments, and unexpected debris left by human civilization below. Far abyssal seafloor. The deepest ocean contains pollution, including plastics and chemicals, transported from distant surface activity. Our abyssal abyss. Scientists discovered plastic fibers and persistent toxins inside trench animals, showing contamination reaches astonishing depths. Our abyssal silence. That unsettling finding turns the trench into a mirror, reflecting humanity's planetary footprint quite clearly. Our abyssal worlds. Mapping the trench relies on multi-beam sonar, which sends sound pulses and times their echoes. Our abyssal shadows. Because sound travels differently with temperature, salinity, and pressure calibration is crucial for accuracy today. Our abyssal frontiers. Autonomous landers drop instruments down record data, then release ballast and float back upward today. Our sinking depths. Remotely operated vehicles can film details, though extreme depths push cables, electronics, and power systems. Our sinking waters. Pressure housings protect cameras and computers, but every seal becomes critical in HADL exploration design. Our sinking pressure. Engineers test materials relentlessly because microscopic flaws can become catastrophic failures under such crushing stress. Our sinking darkness. Water itself changes behavior under pressure, influencing chemistry and sound propagation and biological processes below today. Our sinking trenches. Even simple experiments become difficult when collection, storage, and decompression threaten delicate deep organisms today. Our sinking currents. Some scientists build pressure-retaining samplers so specimens reach laboratories without losing their native conditions today. Our sinking seafloor. These tools help reveal enzymes, microbes, and tissues that function best under pressures unimaginable topside. Our sinking abyss. Challenger Deep is not one pit. Surveys suggest multiple basins and complex topographic structures today. Our sinking silence. Steep walls descend toward comparatively flatter floors covered by fine sediments settling through dark water. Our sinking worlds. Submarine landslides and earthquakes can reshape slopes, redistributing mud and exposing fresh rock surfaces today. Our sinking shadows. That constant remodeling means the trench floor is dynamic and not a static, forgotten landscape today. Our sinking frontiers. Organic material sinks slowly from above, taking days or weeks to reach lower depths today. Our midnight depths. Anything falling there enters a quiet conveyor of decay, scavenging, and bacterial recycling below today. Our midnight waters. Large carcasses attract scavengers fast because opportunities are rare in such nutrient-poor environments today there. Our midnight pressure. Amphipods can swarm baited traps quickly, revealing how efficiently deep scavengers detect food sources today. Our midnight darkness. Scientists use landers with bait to observe animals otherwise difficult to locate across vast darkness. Our midnight trenches. Cameras may sit for hours before motion appears, then suddenly fill with curious feeding creatures. Our midnight currents. Many trench animals appear pale because camouflage against light matters little in perpetual darkness below. Our midnight seafloor. Some species still possess eyes, while others reduce them and rely on different senses today. Our midnight abyss. Chemosensory abilities help detect food mates or predators in a world without sunlight cues today. Our midnight worlds. Scientists study these patterns to understand communication geology and how the ocean transmits energy today. Our midnight shadows. Deep currents also matter, moving oxygen and fine particles through terrain that seems perfectly still. Our midnight frontiers. Although motion looks slow, water continually circulates, linking trenches to broader ocean systems above today. Our crushing depths. Oxygen reaches HADL depths because cold polar waters sink and eventually ventilate the deep ocean. Our crushing waters. Without that global circulation complex, trench animals would struggle to survive below the waves today. Our crushing pressure. The trench helps scientists test limits of life, refining ideas about habitability on other worlds. Our crushing darkness. If organisms endure crushing pressure here, perhaps alien oceans might also host resilient living systems. Our crushing trenches. Moons like Europa or Enceladus inspire comparisons, though their chemistry and energy sources differ significantly. Our crushing currents. Still, Mariana research sharpens tools and questions for future astrobiology missions beyond Earth today there. Our crushing seafloor. The trench also informs materials science, robotics, and sensor engineering built for extreme environments today.
Our crushing abyss. Every successful dive combines geology, biology, physics, chemistry, navigation, and remarkable human persistence today there.
Our crushing silence. Contrary to legend, Our crushing worlds. Instead, the real wonder is how ordinary-seeming organisms persist where pressure should overwhelm fragile biology. Our crushing shadows. Scientists still debate depth limits for fish because pressure eventually constrains vertebrate cellular function today. Our crushing frontiers. Hadal snailfish seem near that boundary, thriving deeper than most other known fishes on Earth. Our Pacific depths. Below certain depths, invertebrates dominate, showing different body plans can handle greater compression effectively today. Our Pacific waters. Sediment microbes may be especially important, driving unseen biochemical cycles across the trench floor today. Our Pacific pressure. These microbial communities break down detritus, transform chemicals, and support wider ecosystems indirectly today there. Our Pacific darkness. Some trenches may receive carbon rapidly during quakes, burying organic matter inside deep sediment pockets. Far Pacific trenches. That means trenches could influence the long-term carbon cycle more than once assumed today there. Far Pacific currents. Researchers continue quantifying how much carbon enters, transforms, and remains locked within Hadal sediments today. Far Pacific seafloor. Mariana system includes not only Challenger Deep, but also long trench segments and neighboring depths. Far Pacific abyss. Different sections vary in slope, sediment thickness, and seismic activity and community composition today there now. Far Pacific silence. Sampling multiple sites prevents scientists from mistaking one unusual location for the entire trench today. Far Pacific worlds. Better maps also reveal small ridges and scarps that shape habitats for deep organisms today. Far Pacific shadows. Exploration remains expensive, so every expedition must balance science goals, risk, and limited time today. Far Pacific frontiers. Weather at the surface still matters because ships support dives, launches, and recoveries today there. Far oceanic depths. A storm above can halt work even when the target lies kilometers beneath calmer darkness. Far oceanic waters. That contrast emphasizes how deep exploration depends on both ocean engineering and surface logistics today. Far oceanic pressure. Public fascination often focuses on depth alone, yet trench matters because it connects many sciences. Far oceanic darkness. It tells a story about plate motion, adaptation, pollution discovery, and Earth's hidden circulation today. Far oceanic trenches. Few people will ever visit, but trench knowledge still changes how humanity understands the planet. Far oceanic currents. Students learn scale through the trench because familiar mountains seem smaller beside such ocean depth. Far oceanic seafloor. If Everest stood inside Challenger Deep, its summit would still remain underwater by a mile. Far oceanic abyss. That comparison is imperfect but useful, helping audiences imagine otherwise abstract measurements of depth today. Far oceanic silence. Another misconception says pressure instantly crushes everything, yet submersibles survive through careful engineering choices today. Far oceanic worlds. Sphere syntactic foam titanium and meticulous testing allow descent into places once deemed unreachable today. Far oceanic shadows. Likewise, trench animals are not primitive leftovers. They are highly adapted modern organisms today there. Far oceanic frontiers. Evolution never stopped there, and it kept sculpting bodies, behaviors, and chemistry for Hadal survival today. Far endless depths. Each expedition raises fresh questions about biodiversity, reproduction, food webs, and geological change below today. Far endless waters. Scientists hope long-term instruments will reveal seasonal patterns hidden by short human visits today there. Far endless pressure. They also want clearer answers about noise pollution and climate-linked changes reaching extreme depths today. Far endless darkness. Because the deep ocean is connected, surface choices eventually influence even the Mariana Trench today. Far endless trenches. Protecting ocean health therefore includes places nobody sees, from coral reefs to Hadal mud today. Far endless currents. The trench is remote, but its discoveries belong to everyone sharing one dynamic planet today. Far endless seafloor. Standing on a ship above it, you see waves, only not the abyss beneath today.
Far endless abyss. Yet below ordinary surface lies a world as alien and instructive as anywhere on Earth.
Far endless silence. Mariana Trench reminds us the planet remains partly unexplored, even in an age of satellites. Far endless worlds. Its darkness hides not fantasy kingdoms, but real science and real life and real planetary history. Far endless shadows. That combination of mystery and evidence is why the deepest ocean keeps pulling explorers downward. Far endless frontiers, and every descent returns humbling message: Earth is stranger and deeper and more connected than expected. Beneath Antarctic sea ice, saltier water can sink like an invisible river and freeze upward from below. Unlike ordinary icicles, a brinacle grows downward underwater around a falling stream of extremely cold brine. Seawater ice rejects salt as crystals form, concentrating leftover liquid inside microscopic pockets between crystals. Those pockets connect into channels, creating hidden plumbing that drains supercold brine from the ice ceiling. Glassy ice. When dense brine escapes, it sinks because extra salt makes it heavier than surrounding seawater below. Hidden ice. The escaping liquid is colder than nearby seawater, so water touching it can freeze almost immediately. Winter ice. Ice forms around the plume, building a hollow tube, while brine continues flowing through its center. That tube is a brinacle, sometimes called an underwater icicle, though its mechanism is reversed. Frigid polar ice. Calm water lets the fragile tube extend downward. Currents can break it before reaching the seafloor. Shallow ice. If tube touches bottom, brine spreads sideways and can freeze a creeping path across sediment-distant ice. Small, slow animals may become trapped when brine pools and new ice surround them locally. Crystal polar ice.
This icy finger is not lava, poison, or magic. It is physics driven by
surface freezing begins the process by forcing dissolved salt out of growing ice crystals above. Ancient polar ice. Fresh ice becomes a porous solid, while rejected salt creates dense liquid trapped within gaps. Narrow polar ice. As winter continues, these salty pockets grow colder and more concentrated inside the sea ice matrix. Gravity pulls the densest brine downward through channels whenever a connected pathway opens beneath ice. Dense polar ice. The brine remains liquid because high salinity lowers its freezing point below surrounding seawater's temperature. Briny polar ice. Surrounding seawater freezes at the brine boundary, forming a shell rather than freezing everything nearby. A narrow shell focuses the flow, allowing the tube to grow longer as brine drains. Frozen polar ice. The structure is hollow because the coldest salty liquid keeps moving through the middle continuously. Early brinacle walls are delicate and transparent, easily broken by currents, animals, or slight contact. Clear polar ice. The tube length depends on brine supply, water depth, currents, temperature, and seafloor distance. A shallow, calm seafloor gives the descending tube enough time to reach the bottom intact. Beneath deep polar ice. Deep water often prevents contact because the brinacle breaks, melts, or exhausts its supply beneath local polar ice. When the brine plume stops, tube growth ends and surrounding seawater gradually erodes the ice. Cold polar ice.
The phenomenon looks like frozen lightning, but every branch follows local flow and freezing conditions. Fresh polar ice.
A brinacle grows opposite a stalactite because cold liquid, not cold air, builds the tube. Salty polar ice. Inside the tube, brine can keep sinking while the outer ice thickens around it. Beneath bright polar ice.
Tide leaks may create branches, but most flow usually continues downward along the easiest path. Dark polar ice.
The growing finger records a moving boundary between dense brine and less salty seawater. Beneath still polar ice. At the bottom, the heavy brine hugs sediment because density keeps it below ordinary seawater. Gentle polar ice. A starfish moves slowly enough that changing chemistry and ice can overtake its escape. Beneath thin polar ice. Faster animals may avoid the path because the hazard is localized and physically constrained. Beneath hollow polar ice. The danger comes from extreme cold and salinity, not from a venom or infection. Beneath layered polar ice. Bottom depressions collect brine, forming temporary pools with conditions unlike the water above them. Those pools may persist while supply continues, then dilute when currents or mixing return. Beneath slow polar ice, brinicles are spectacular, but they require a narrow window of polar conditions to survive. Beneath stable polar ice, moving ice, strong currents, or warm water can destroy the tube before it matures. Beneath broken polar ice, frozen path is therefore a local event, not an ocean-wide freezing chain reaction. Beneath temporary polar ice, scientists recreate brinicles in cold tanks by letting dense brine drain into near-freezing seawater beneath curved polar ice.
Laboratory tanks reveal the plume first, then the icy wall growing around its boundary. Beneath branching polar ice,
Older brine can build faster tubes because it removes heat from surrounding water more effectively. Smooth polar ice. Salinity controls density, so the brine falls instead of floating back toward the ice. Beneath low polar ice, flow speed matters because rapid drainage can stretch the tube before walls become strong. Beneath heavy polar ice, if flow becomes too weak, new ice stops forming and the existing tube begins fading. Falling polar ice. A brinicle behaves like a self-building pipe assembled by the fluid running inside it. Beneath sinking polar ice, the pipe's wall grows wherever seawater crosses the freezing boundary near the descending plume. Beneath rising polar ice, this makes brinicles useful examples of self-assembly in cold fluid systems on Earth. Beneath porous polar sea ice, their geometry resembles chemical gardens, except the dense internal solution grows downward instead of upward. Watery polar ice. Osmotic differences can help feed brine drainage by drawing seawater through porous ice structures. Beneath icy polar ice, pressure then releases concentrated brine through weak spots, restarting downward tube formation beneath ice. Beneath marine polar ice, yet small differences in temperature, salinity, and turbulence decide whether a brinicle appears. Beneath Antarctic polar sea ice. Sea ice is not solid glass. It contains liquid pathways, crystals, bubbles, and living microbes. Those hidden pathways shape polar ecosystems by moving salt, nutrients, gases, and organisms between layers. Coastal polar ice. Brinicles are one visible outcome of the same brine rejection that helps form dense polar waters. Benthic ice. At ocean scale, sinking salty water contributes to circulation, but brinicles are tiny local structures, blue-green polar ice. The thumbnail version is dramatic. The scientific version is delicate, conditional, and deeply physical. Divers have observed brinicles as fragile glassy fingers beneath sea ice in polar environments, beneath teal polar ice. Their beauty can be misleading because a thin, transparent tube hides very cold, dense liquid. Violet polar ice. Touching one may break it, proving the structure is delicate despite its seafloor effects. Beneath amber polar ice. The brine inside can continue downward, even if the outer shell cracks or branches. Beneath elegant polar ice. Sailing nearby water shows sharp differences between ordinary seawater and the descending internal plume. Beneath remote polar ice. Cameras capture growth by watching where new ice appears around the invisible temperature boundary. Beneath delicate polar ice. Because the process has no flames, visual cues must show salinity, density, and freezing instead. A clear cutaway helps viewers understand why the tube is hollow rather than solid. Beneath measured polar ice. Without readable labels, shapes can still show salt rejection, sinking flow, and ice formation. The brinicle's power comes from phase change, where liquid water reorganizes into solid ice. Beneath mottled polar ice. Seafloor life near a brinicle faces rapid environmental change in a very small area. Beneath natural polar ice. Animals adapted to cold water can still be overwhelmed by brine that is colder and saltier. Living ice. A brine path may trap slow echinoderms, while nearby open water remains survivable for others. Quietest polar ice, and this selective impact makes brinicles ecological microevents rather than universal death machines below ice. Beneath coldest polar ice. The white trail is not an infection spreading. It is freezing following dense bottom flow, saltiest polar ice. Local topography matters because brine seeks low areas, channels, hollows, and gentle slopes. Beneath nearest polar sea ice, when supply ends, ordinary mixing can dilute brine pools and erase the frozen pattern. Beneath lowest polar ice, seasonal conditions decide whether new sea ice keeps producing enough concentrated brine overhead. Beneath clearest polar sea ice, a single winter night can build structures that later disappear as the environment shifts. Beneath soft polar ice, brinicles therefore reveal polar change through fragile shapes that exist only under specific balances. Planetary scientists study brinicles because icy worlds may contain brines beneath frozen shells, beneath open polar sea ice. On moons like Europa, similar gradients could create self-assembled ice structures in hidden oceans. Beneath closed polar ice forests, those alien possibilities remain hypothetical, while Earth brinicles provide real, observable physical analogs beneath small polar sea ice. Chemical gradients near ice membranes could concentrate molecules and create tiny reactive environments beneath broad polar sea ice. Such ideas connect polar ocean physics with broader questions about chemistry in icy habitats. Beneath complex polar ice, the analogy should stay cautious because extraterrestrial oceans differ greatly from Antarctic seawater. Still, brinicles show how freezing, salt, and gravity can build complex structures without life. Beneath rare polar ice, they also show that simple ingredients can create dramatic patterns when conditions are precisely balanced. Common polar ice. A good visual explanation follows energy and matter, not just the strange final shape. Beneath exact polar ice,
the chain begins with freezing sea ice and ends with a cold river along the seafloor. Visible ice.
Every step preserves the same rule. Salt changes freezing point, density, and motion together. That rule makes the lake-like ocean below sea ice freeze from beneath in narrow paths. Scientific polar ice. A brinicle is therefore a frozen record of sinking brine, not a creature or curse. Cinematic polar ice. Its growth can appear alive because the tube extends, branches, and searches downward through water. Balanced polar ice. But each motion follows fluid mechanics, heat transfer, and crystal growth at the boundary, beneath precise polar ice. The most cinematic part is also the most scientific. Invisible brine drawing visible ice around itself. Extreme ice. For viewers, the key is separating spectacle from mechanism while keeping the danger understandable. Beneath patient polar ice. The icy finger forms only where source, temperature, salinity, depth, and calm water align. Remove any one ingredient, and the brinicle may fail before touching the seabed. Beneath clean polar sea ice, the under-ice scene becomes less mysterious once density explains why the cold river sinks. Freezing explains the walls, salinity explains the motion, and calm water explains the shape, beneath filtered polar ice. Together, these processes turn a hidden brine leak into a downward-growing tube of ice, beneath microbial polar ice. The seafloor trail shows what happens after the tube delivers dense brine to bottom terrain. Planetary polar ice. A spreading brine pool can outline the invisible flow using freshly formed ice as evidence. Analog polar ice. Under the sea ice, the world is not frozen solid. It is chemically layered. Beneath field polar ice, brinicles expose those layers by making a descending salty current visible as growing ice. Beneath laboratory polar ice. The final image returns to the first droplet because the cycle begins whenever brine drains. Ecological polar ice. One falling stream can become a tube, a trail, and a lesson about polar physics. Mechanical polar ice. The frozen river beneath the ice is really salt, gravity, and heat moving together. Beneath thermal polar ice. A brinicle begins quietly, grows delicately, and can reshape a small patch of seafloor. Beneath osmotic polar ice. The strange underwater icicle teaches that freezing can flow downward when salt changes water's rules. Saline polar ice. What looks supernatural is a precise interaction between rejected salt and water near its freezing point. Crystalline ice. The Brinicle's path marks where invisible density currents made the ocean briefly build ice. Beneath brackish polar ice. From ceiling to seafloor, the whole event is an elegant cascade of cold brine and crystal growth, translucently. When the final droplet falls, the underwater river starts again from the frozen ceiling above. Low-flow polar ice. Another drainage pathway can start nearby, proving brinicles are repeated events rather than single anomalies. Stillwater polar ice. Each new tube depends on the same interaction between rejected salt, dense flow, and freezing seawater. Icebound ice. The polar ocean keeps most water liquid, while narrow brine rivers draw ice around themselves. Underside polar ice. Endlessly repeated freezing and rejection can restart the silent underwater river whenever winter conditions return. Seafloor polar ice. Silent shoreline frame shows amber water preserving rusted animals through indigo minerals, not instant stone magic here quietly. Silent shoreline view explains basalt brine concentrating saffron birds as jagged salts builds bright legends around vivid shallows. Silent shoreline moment reveals cerulean shore surviving turquoise bats because kelp crystals supports cold myths despite white islands. Silent shoreline cutaway traces desert basin entering umber feathers while limestone evaporation leaves dry hazards across cracked mudflats. Silent shoreline motion contrasts Ember Lake with velvet remains so misty carbonates separates electric chemistry from dark springs. Silent shoreline detail follows frosted spring forming willow bodies after nocturnal natron exposes fragile ecology beneath exposed wetlands. Silent shoreline contrast shows golden mud preserving xenial bones through opal trona, not rocks swallowing living animals quietly. Silent shoreline sequence explains halite crust concentrating yellow wings as porous alkalinity builds hot toxicity around gentle basins. Silent shoreline evidence reveals ivory surface surviving zircon tracks because quiet drying supports icy refuge despite hazard channels. Silent shoreline pattern traces jade wetland entering ashen nests, while red coating leaves layered survival across isolated margins. Silent shoreline signal contrasts kaolin margin with brassy flocks, so silica brine separates muted death from low sediments. Silent shoreline turn follows lunar flat forming cobalt chicks after tawny sunlight exposes narrow preservation beneath mirrored valleys. Silent crater frame shows marble plume preserving dusky fish through ultra wind, not volcano fire beneath water quietly. Silent crater view explains nickel pool concentrating earthen microbes as violet silt builds pale sampling around open colonies. Silent crater moment reveals ochre mirror surviving faint samples because weathered films supports rough caution despite powdered surfaces. Silent crater cutaway traces pearl water entering glassy animals, while young minerals leave silver evidence across remote shorelines. Silent crater motion contrasts quartz brine with hollow birds, so zephyr salts separates thin legends from salted shallows. Silent crater detail follows rusted shore forming indigo bats after ancient crystals exposes uneven myths beneath turbid islands. Silent crater contrast shows saffron basin preserving jagged feathers through bright evaporation, not safe swimming conditions nearby quietly.
Silent crater sequence explains turquoise lake concentrating kelp remains as cold carbonates builds white chemistry around warm springs.
Silent crater evidence reveals umber springs surviving limestone bodies because dry natron supports cracked ecology despite arid wetlands. Silent crater pattern traces velvet mud entering misty bones, while electric trona leaves dark adaptation across bleached crusts. Silent crater signal contrasts willow crust with nocturnal wings, so fragile alkalinity separates exposed toxicity from clear basins. Silent crater turn follows xenial surface forming opal tracks after granular drying exposes fine refuge beneath dense channels. Silent lagoon frame shows yellow wetland preserving porous nests through hot coating, not death happening on contact quietly. Silent lagoon view explains zircon margin concentrating quiet flocks as icy brine builds hazard death around flat sediments. Silent lagoon moment reveals ashen flat surviving red chicks because layered sunlight supports isolated preservation despite ghost valleys. Silent lagoon cutaway traces brassy plume entering silica fish, while muted wind leaves low color across heavy reflections. Silent lagoon motion contrasts cobalt pool with tawny microbes so narrow silt separates mirrored sampling from inland colonies. Silent lagoon detail follows dusky mirror forming ultrasamples after oxidized films exposes neutral caution beneath lacustrine surfaces. Silent lagoon contrast shows earth and water preserving violet animals through pale minerals, not every bloom becoming poisonous quietly. Silent lagoon sequence explains faint brine concentrating weathered birds as rough salts builds powdered legends around pinkish shallows. Silent lagoon evidence reveals glassy shore surviving young bats because silver crystals supports remote myths despite shallow islands. Silent lagoon pattern traces hollow basin entering zephyr feathers, while thin evaporation leaves salted hazards across alkaline mudflats. Silent lagoon signal contrasts indigo lake with ancient remains, so uneven carbonate separates turbid chemistry from briny springs. Silent lagoon turn follows jagged spring forming bright bodies after vivid natron exposes unstable ecology beneath caustic wetlands. Silent basin frame shows kelp mud preserving cold bones through white trona, not alkali acting like lava quietly. Silent basin view explains limestone crust concentrating dry wings as cracked alkalinity builds arid toxicity around evaporite basins. Silent basin moment reveals misty surface surviving electric tracks, because dark drying supports bleached refuge despite rift channels. Silent basin cutaway traces nocturnal wetland entering fragile nests, while exposed coating leaves clear survival across soda margins. Silent basin motion contrasts opal margin with granular flocks so fine brine separates dense death from trona sediments. Silent basin detail follows porous flat forming hot chicks after gentle sunlight exposes eroded preservation beneath natron valleys. Silent basin contrast shows quiet plume preserving icy fish through hazard wind, not myth overpowering ecology facts quietly. Silent basin sequence explains red pool concentrating layered microbes as isolated silt builds ghost sampling around thermal colonies. Silent basin evidence reveals silica mirror surviving muted samples because low film supports heavy caution despite carbonate surfaces. Silent basin pattern traces tawny water entering narrow animals while mirrored minerals leaves inland evidence across muddy shorelines. Silent basin signal contrasts ultrabrine with oxidized birds, so neutral salt separates lacustrine legends from crimson shallows. Silent basin turn follows violet shore forming pale bats after open crystals exposes mineral myths beneath fiery islands. Silent surface frame shows weathered basin preserving rough feathers through powdered evaporation, not bones changing into granite quietly. Silent surface view explains young lake concentrating silver remains as remote carbonates builds shallow chemistry around spectral springs. Silent surface moment reveals zephyr springs surviving thin bodies because salted natron supports alkaline ecology despite living wetlands. Silent surface cutaway traces ancient mud entering uneven bones, while turbid trona leaves briny adaptation across final crusts. Silent surface motion contrasts bright crust with vivid wings, so unstable alkalinity separates caustic toxicity from amber basins. Silent surface detail follows cold surface forming white tracks after warm drying exposes closed refuge beneath basalt channels. Silent surface contrast shows dry wetland preserving cracked nests through arid coating, not lake swallowing all wildlife quietly. Silent surface sequence explains electric margin concentrating dark flocks as bleached brine builds rift death around desert sediments. Silent surface evidence reveals fragile flat surviving exposed chicks because clear sunlight supports soda preservation despite ember valleys. Silent surface pattern traces granular plume entering fine fish, while dense wind leaves trona color across frosted reflections. Silent surface signal contrasts hot pool with gentle microbes, so eroded silt separates natron sampling from golden colonies. Silent surface turn follows icy mirror forming hazard samples after flat films exposes volcanic caution beneath halite surfaces. Silent mudflat frame shows layered water preserving isolated animals through ghost minerals, not one lake explaining all quietly. Silent mudflat view explains muted brine concentrating low birds as heavy salts builds carbonate legends around jade shallows. Silent mudflat moment reveals narrow shore surviving mirrored bats because inland crystals supports muddy myths despite kaolin islands. Silent mudflat cutaway traces oxidized basin entering neutral feathers while lacustrine evaporation leaves crimson hazards across lunar mudflats.
Silent mudflat motion contrasts pale lake with open remains, so mineral carbonate separates fiery chemistry from marble springs.
Silent mudflat detail follows rough spring forming powdered bodies after pinkish natron exposes silent ecology beneath nickel wetlands. Silent mudflat contrast shows silver mud preserving remote bones through shallow trona, not adaptation cancelling every risk quietly. Silent mudflat sequence explains thin crust concentrating salted wings as alkaline alkalinity builds living toxicity around pearl basins. Silent mudflat evidence reveals uneven surface surviving turbid tracks because briny drying supports final refuge despite quartz channels.
Silent mudflat pattern traces vivid wetland entering unstable nests, while caustic coating leaves amber survival across rusted margins.
Silent mudflat signal contrasts white margin with warm flocks, so closed brine separates basalt death from saffron sediments. Silent mudflat turn follows cracked flat forming arid chicks after evaporite sunlight exposes cerulean preservation beneath turquoise valleys. Silent spring frame shows dark plume preserving bleached fish through rift wind, not brine acting like glue quietly. Silent spring view explains exposed pool concentrating clear microbes as soda silt builds ember sampling around velvet colonies. Silent spring moment reveals fine mirror surviving dense samples because tronafilms supports frosted caution despite willow surfaces. Silent spring cutaway traces gentle water entering eroded animals, while natron minerals leaves golden evidence across xenial shorelines. Silent spring motion contrasts hazard brine with flat birds, so volcanic salt separates halite legends from yellow shallows. Silent spring detail follows isolated shore forming ghost bats after thermal crystals exposes ivory myths beneath zircon islands. Silent spring contrast shows low basin preserving heavy feathers through carbonate evaporation, not algae thriving without salts quietly. Silent spring sequence explains mirrored lake concentrating inland remains as muddy carbonates builds kaolin chemistry around brassy springs. Silent spring evidence reveals neutral springs surviving lacustrine bodies because crimson natron supports lunar ecology despite cobalt wetlands. Silent spring pattern traces open mud entering mineral bones, while fiery trona leaves marble adaptation across dusky crusts. Silent spring signal contrasts powdered crust with pinkish wings, so silent alkalinity separates nickel toxicity from earthen basins. Silent spring turn follows remote surface forming shallow tracks after spectral drying exposes ochre refuge beneath faint channels. Silent desert frame shows salted wetland preserving alkaline nests through living coating, not stone bodies walking later quietly. Silent desert view explains turbid margin concentrating briny flocks as final brine builds quartz death around hollow sediments.
Silent desert moment reveals unstable flat surviving caustic chicks because amber sunlight supports rusted preservation despite indigo valleys.
Silent desert cutaway traces warm plume entering closed fish, while basalt wind leaves saffron color across jagged reflections. Silent desert motion contrasts arid pool with evaporite microbes, so cerulean silt separates turquoise sampling from kelp colonies. Silent desert detail follows bleached mirror forming rift samples after desert films exposes umber caution beneath limestone surfaces. Silent desert contrast shows clear water preserving soda animals through ember minerals, not evidence cards needing words quietly. Silent desert sequence explains dense brine concentrating trona birds as frosted salts builds willow legends around nocturnal shallows. Silent desert pattern traces flat basin entering volcanic feathers, while halite evaporation leaves yellow hazards across porous mudflats. Silent desert signal contrasts ghost lake with thermal remains, so ivory carbonates separates zircon chemistry from quiet springs. Silent margin frame shows inland mud preserving muddy bones through kaolin trona, not red pigments being blood quietly. Silent margin view explains lacustrine crust concentrating crimson wings as lunar alkalinity builds cobalt toxicity around tawny basins. Silent margin moment reveals mineral surface surviving fiery tracks because marble drying supports dusky refuge despite ultra channels. Silent margin cutaway traces pinkish wetland entering silent nests, while nickel coating leaves earthen survival across violet margins. Silent margin motion contrasts shallow margin with spectral flocks, so ochre brine separates faint death from weathered sediments. Silent margin detail follows alkaline flat forming living chicks after pearl sunlight exposes glassy preservation beneath young valleys. Silent margin contrast shows briny plume preserving final fish through quartz wind, not beauty, hiding no hazard quietly. Silent margin sequence explains caustic pool concentrating amber microbes as rusted silt builds indigo sampling around ancient colonies. Silent margin evidence reveals closed mirror surviving basalt samples because saffron film supports jagged caution despite bright surfaces. Silent margin pattern traces evaporite water entering cerulean animals while turquoise minerals leaves kelp evidence across cold shorelines. Silent margin signal contrasts rift brine with desert birds so umber salts separates limestone legends from dry shallows. Silent margin turn follows soda shore forming ember bats after velvet crystals exposes misty myths beneath electric islands.
Silent mirror frame shows Trona Basin preserving frosted feathers through willow evaporation, not all myths being useless quietly.
Silent mirror view explains Natron Lake concentrating golden remains as xenial carbonates builds opal chemistry around granular springs. Silent mirror moment reveals volcanic springs surviving halite bodies because yellow natron supports porous ecology despite hot wetlands. Silent mirror cutaway traces thermal mud entering ivory bones, while zircon trona leaves quiet adaptation across icy crusts. Silent mirror motion contrasts carbonate crust with jade wings, so ashen alkalinity separates red toxicity from layered basins.
Silent mirror detail follows muddy surface forming kaolin tracks after brassy drying exposes silica refuge beneath muted channels.
Silent mirror contrast shows crimson wetland preserving lunar nests through cobalt coating, not lava turning bodies solid quietly. Silent mirror sequence explains fiery margin concentrating marble flocks as dusky brine builds ultra-death around oxidized sediments. Silent mirror evidence reveals silent flat surviving nickel chicks because earthen sunlight supports violet preservation despite pale valleys. Silent mirror pattern traces spectral plume entering okra fish, while faint wind leaves weathered color across rough reflections. Silent mirror signal contrasts living pool with pearl microbes, so glassy silt separates young sampling from silver colonies. Silent mirror turn follows final mirror forming quartz samples after hollow films exposes zephyr caution beneath thin surfaces.
This South American fish can activate another animal's muscles without touching it, using electricity generated inside its own body. Electric eels inhabit slow rivers, floodplains, and seasonally changing waters across northern South America, especially within quiet backwaters. The habitats are dark, cluttered, and difficult to search visually, making electrical sensing far more useful than eyesight. Electric sensing turns murky water into a readable electrical landscape. The advantage matters during dark water navigation. Despite its common name, an electric eel is a South American knife fish rather than a true eel.
A long anal fin provides smooth forward, backward, and hovering movement, while the elongated body remains nearly straight. Precise hovering keeps the sensory field aimed steadily, while the animal investigates prey hidden among roots and leaf litter.
Electric eels regularly reach the surface because atmospheric air supplies most of the oxygen supporting their metabolism.
A highly vascular mouth lining absorbs oxygen after every surface gulp. Control strengthens during fast predator escape. Air breathing permits survival in stagnant pools, where dissolved oxygen can fall below levels tolerated by many fishes. Most of the elongated body behind the vital organs contains specialized tissues devoted to generating electrical discharges.
Electrocytes are specialized cells arranged so their tiny voltages add together. That helps during muscle cell transformation.
Evolution transformed muscle-derived cells by reducing contraction, expanding membrane specialization, and coordinating their electrical activation together. Asymmetric nerve input lets each electrocyte create a directional voltage difference. Control strengthens during synchronized neural firing. A neural command opens ion channels almost simultaneously across the organ. Efficiency rises during sodium channel opening. Each activated cell contributes only a small electrical potential. Thousands combine to create the organ's full voltage.
Series arrangement adds the tiny voltage from each cell, producing a much larger discharge across the complete organ.
Parallel arrangement increases available current, while series arrangement raises total voltage. That helps during parallel current amplification.
Surrounding water completes the external circuit whenever a high voltage pulse travels from the eel toward another animal.
Genetic and anatomical evidence now separates electric eels into 3 living species, notably during species-level divergence. Each recognized species occupies a different combination of regional geology, water conductivity, and evolutionary history across Greater Amazonia. Electrical performance changes with the conductivity of surrounding water. Conductivity reshapes both current pathways and target exposure. Water chemistry reshapes the current pathway, changing how strongly a discharge crosses prey, predators, and surrounding habitat. Voltage alone cannot describe the biological effect of an electric discharge. Performance improves during multi-factor electrical effects.
Current, pulse duration, conductivity, and contact geometry together determine the biological effect of any electrical discharge underwater.
During a powerful pulse, the eel's head and tail behave as opposite electrical poles within the surrounding water.
Body position lets the animal shape where the strongest current travels. Precision increases during body position control.
Weaker pulses serve continuous sensing and communication, while powerful volleys are reserved mainly for predation and defense.
Objects with different conductivity distort the eel's self-generated field, allowing nearby structures to be detected without vision. Electroreceptors distributed across the skin sample tiny field changes created by nearby objects, prey, and environmental boundaries.
Electrical distortions become spatial information inside the nervous system. That neural map guides movement through cluttered darkness.
Active electrolocation reveals obstacles even when vision contributes almost nothing. The advantage matters during obstacle shape reconstruction. Living prey produces a distinctive electrical signature compared with stones, wood, sediment, and empty surrounding water nearby. Movement changes the pattern repeatedly, helping the eel separate prey from static background clutter during prey tracking. Pulse timing can change with context, arousal, and nearby electrical information. That helps during context-dependent pulsing. Weak electric discharges also carry social information between individuals without triggering a predatory or defensive attack underwater. Electrical signals can help distinguish species, individuals, and behavioral states. Performance improves markedly during species signal recognition. After detecting prey, the electrical system rapidly shifts from environmental mapping toward high-voltage muscular control instantly. A hunting eel can activate nearby prey motor neurons remotely, forcing muscles to respond before physical contact occurs. The prey's own nervous system carries the eel's externally imposed command. This matters during prey nerve firing. High-frequency volleys force simultaneous contraction across the prey's body, producing a brief period of whole-body muscular tetanus. A temporary tetanus blocks coordinated escape during the eel's final approach through cluttered, low-visibility habitat underwater.
Controlled experiments reproduce the same muscular response by applying eel-like electrical stimulation directly to isolated prey preparations.
Closely spaced pulses prevent full relaxation between twitches, merging separate contractions into one sustained, forceful muscular response.
Prey becomes immobilized because its own nerves and muscles obey externally imposed electrical stimulation from the eel.
Electricity does not physically push the target. Instead, it recruits the target's motor neurons and muscle fibers.
External voltage crosses the threshold needed to initiate action potentials. That becomes valuable during motor threshold crossing. Neural activation closes the timing gap between detection and capture. Control remains useful during rapid neural conduction.
A sudden high-voltage burst can interrupt an escape turn before the prey completes the movement or accelerates away. Immobilization creates a short capture window, allowing the eel to close distance before coordinated swimming can resume.
Curling positions both electrical poles near the target. This bioelectric mechanism remains important during opposite pole positioning.
This posture greatly increases current through the prey's body. Curling therefore amplifies stimulation without raising internal output.
Curling strengthens stimulation through geometry, increasing current across the prey without raising the organ's internal voltage further.
Repeated volleys keep struggling prey from regaining coordinated movement. Continued stimulation preserves the eel's brief capture advantage. High-voltage hunting carries a metabolic and electrical cost. Recovery between attacks restores ion gradients across electrocyte membranes.
Between attacks, membrane pumps rebuild sodium and potassium gradients across thousands of electrocytes inside the electric organs.
Recharge depends on active transport, resetting thousands of biological battery cells. This matters during ion pump resetting. When prey remains hidden, the eel emits brief electrical doublets that can force an involuntary muscle twitch from concealment. That twitch moves leaves, sediment, or water, revealing the hidden animal against an otherwise motionless underwater background.
Laboratory doublets reproduce the same rapid muscular twitch observed when eels searched naturally for concealed prey underwater. Spacing between two pulses is well suited to summating the prey's muscle response into one detectable movement.
After delivering a doublet, the predator pauses briefly and monitors the surrounding electric field for nearby motion. Electrolocation converts the involuntary twitch into a precise positional clue. This remains important during twitch signal detection. Multiple viewpoints improve localization inside cluttered cover. That electrical strategy provides major advantages during multi-angle localization. Movement distinguishes living prey from inert debris more reliably than static electrical shape within cluttered underwater habitat.
Once the target is located, the eel immediately replaces probing doublets with an immobilizing high-voltage volley. The same bioelectric system first exposes concealed prey, then prevents that prey from escaping the approaching predator. A complete hunt combines weak electrolocation, forced twitching, high-voltage immobilization, body curling, and rapid physical capture.
Low-voltage discharges continuously refresh the eel's environmental map before any predatory strike becomes necessary inside cluttered water.
An initial field distortion suggests a possible living target, but cannot confirm identity without additional testing underwater. A doublet challenges the hidden object to respond biologically. Any twitch separates living prey from motionless environmental clutter. The resulting motion confirms that excitable tissue is present. The eel immediately converts confirmation into precise targeting. Twitch geometry helps aim the following high-voltage strike. A clearer orientation strengthens the following immobilizing volley.
The attack volley recruits the prey's own motor system, turning ordinary muscle physiology into a temporary restraint.
Electrical control reduces the need for a prolonged physical chase. This remains important during rapid target capture.
Bioelectric hunting becomes especially valuable where darkness, roots, and sediment make ordinary visual pursuits slow or unreliable.
After feeding, the animal returns to low-voltage sensing rather than maintaining energetically costly attack volleys afterward. Electric eels also direct powerful volleys toward large nearby threats that cannot be treated as ordinary prey. During shallow water defense, an eel may approach vertically and rise toward the intruder instead of retreating.
Pressing the chin against a conductive target creates a more effective pathway for electrical current through the threat.
As the eel climbs higher from the water, measured current entering the contacted object rises progressively and dramatically.
Emerging from the water removes competing current pathways, concentrating more of the discharge through the contacted threat. Repeated high-voltage volleys during brief contact can persuade an approaching predator or intruder to withdraw rapidly. Alexander von Humboldt famously described electric eels rising against horses driven into shallow South American water during an early expedition. Modern experiments confirm defensive leaping, although they cannot verify every dramatic detail preserved in Humboldt's 19th century account.
Instrumented trials confirm that climbing contact increases electrical delivery. Measured current rose as the eel climbed higher.
The same electric tissues power both predatory strikes and defensive leaps. That helps during shared organ deployment.
Sax organ contributes weaker pulses used primarily for electrolocation, orientation, and communication rather than forceful attack underwater.
Proteomic comparisons reveal distinct molecular profiles across the main, hunters, and sax electric organs within specialized tissues. Differences in channels, pumps, and structural proteins help each electric organ meet distinct energetic and signaling demands. Electric organs originate from muscle lineage cells during development. That process repurposes muscle precursors into non-contractile specialized electrical cells. During development, muscle precursors lose ordinary contractile machinery and gain the membrane specialization needed for electrical output. Electric organs evolved independently multiple times among fishes. Distant lineages repeatedly modified muscle through similar genetic pathways. Distant electric fish lineages repeatedly reused related genetic pathways while independently transforming muscle into electricity-producing tissue. Engineers examine electrocyte stacking for ideas about soft ion-based power sources, sensors, and bio-inspired materials today.
Bio-inspired research does not mean a living eel functions like a practical battery for ordinary human technology. Volta's electric eels have been observed gathering in groups exceeding 100 individuals near a remote Amazonian lake.
Coordinated movement herds prey into a dense, shallow aggregation. Herding concentrates many targets within one strike zone. Most eels maintain the encircling formation, while a smaller subgroup advances toward the compressed prey school together.
Several hunters then discharge high-voltage volleys nearly simultaneously. Timing several discharges together broadens the affected prey area.
Synchronized volleys can expose, stun, and scatter many small fish concentrated inside the shallow prey aggregation simultaneously. Nearby group members exploit the brief feeding opportunity created after several hunters discharge electricity together during feeding. The sequence can repeat during a single hunting event. Repeated cycles increase feeding efficiency under exceptional conditions. Similar events recorded in different years suggest structured social predation rather than one accidental gathering over time. Local prey density may make cooperative hunting worthwhile. Dense seasonal shoals can reward brief temporary social coordination. Cooperative hunting likely appears only where prey density, shallow water, and local geography make coordination worthwhile for multiple hunters. Group hunting is documented in Volta's electric eel, but should not be generalized across every species, habitat, or population.
Researchers still do not know how individuals coordinate each strike. The advantage matters during group coordination mechanisms.
Weak electrical pulses, body movements, and spatial position remain plausible signals for coordinating group attacks underwater together.
Field electrode arrays could separate individual discharge patterns during group hunts. This matters during field electrode monitoring.
Synchronized measurements may reveal whether attacks are actively coordinated. Combined data can expose timing of individual roles.
Protecting electric eel behavior requires conserving connected floodplains, tributaries, and seasonal channels throughout the greater Amazon basin.
Dams, pollution, and altered flood cycles can disrupt prey movements, breeding habitat, and seasonal hunting opportunities locally.
Healthy river connectivity preserves both electric eel diversity and the ecological conditions supporting their specialized behavior regionally. Together, electrolocation, remote muscle control, hidden prey detection, curling, leaping, and social hunting reveal exceptionally precise bioelectric behavior. Electric eels are living batteries that sense, command, defend, and sometimes hunt together through carefully controlled bioelectricity.
This seabird can sleep while remaining airborne above the open ocean. Long flights inspired a question that behavior alone could not answer. Researchers studied breeding female great frigate birds from a Galapagos colony. A lightweight recorder was attached before each bird returned to flight.
Electrode measured electrical activity from both brain hemispheres.
An accelerometer recorded head motion and turning forces.
A separate GPS unit tracked position and altitude.
Together, the instruments linked brain state with real flight behavior.
Some recorded journeys covered thousands of kilometers without landing. The longest sleep recordings followed birds aloft for about 10 days. Over deep water, the birds had nowhere safe to perch.
Frigate birds avoid prolonged contact with the ocean surface.
Their plumage is less waterproof than that of many seabirds.